Hello everyone, my name is Rahul. In the previous video, we had discussed about green revolution. In this video, we will study about genetically modified organisms. So plants, bacteria, fungi and animals whose genes have been modified or altered by manipulation are called as genetically modified organisms. So genetically modified organisms can be anyone, plants, bacteria, fungi or animals whose genes have been manipulated in any sense are known as GMOs. So a genetically modified plant will be known as a GMO. Now the Green Revolution, it succeeded in almost tripling the food supply, but yet it was not enough to feed the growing human population. So the increased yields, they were mainly due to improved crop varieties and also the use of better management practices such as agrochemicals, fertilizers, pesticides, etc. However, for countries like India, which are still developing, procuring these chemicals is very expensive for the farmers. So, biotechnology, through the use of genetically modified organisms, provides an alternative method to increase the crop yield without heavy dependence on such chemicals. So, we will study about the use of genetically modified plants. Use of genetically modified plants or genetically modified organisms. So any organism whose genes have been manipulated is a GMO. Now the gene manipulation may be addition of a gene, deletion of a gene, changing in the base sequence, changing in the number of base pairs. It could be anything. So the first use is to enhance the nutritional quality of food. To enhance the nutritional quality of food. This enhancement in nutritional quality is also sometimes known as biofortification. Biofortification. It refers to breeding crops with higher levels of nutrients such as vitamins, minerals, proteins, carbohydrates, etc. So this is mostly a means to improve the public health. This is done with the objectives of biofortification or improved nutritional quality. First of all, to provide or improve protein, protein content and quality. Next is to improve oil content and quality. To improve vitamin content and to improve micronutrient and mineral content of food. Now, some very famous examples are golden rice, in which vitamin A is present in good quantity. So what we did was a gene for production of beta carotene, which is the precursor of vitamin A, was transferred to rice plant. So we took the gene for beta carotene production from those crops which naturally have good vitamin A, such as carrots, and we placed that gene, we transferred that gene to the rice, therefore producing golden rice which has good vitamin A content. Now, not everyone can afford to have salad with their food, but rice, being the staple diet in most of the parts of India, is a good source of vitamin A. In 2000, improved varieties improved varieties of maize were introduced that had twice the amount of amino acids, lysine and tryptophan were introduced. Similarly, a variety of wheat called as Atlas 66 called as Atlas 66 was introduced, which has higher protein content. Next is iron fortified rice. This contains approximately 5 times the iron content of commonly consumed varieties. Also, the Indian Agricultural Research Institute, New Delhi, it released several vegetable crops rich in vitamins and minerals. Vegetable crops rich in vitamins and minerals such as vitamin A enriched carrots, spinach, and pumpkin, vitamin C enriched bitter gourd, bathua, mustard, and tomato, iron and calcium enriched spinach and bathua, and protein enriched beans, which were broad beans, lab beans, French beans, and garden peas and garden peas. So these are all examples of varieties which have been improved to enhance the nutritional quality of food. Now, the next use of genetically modified plants is to reduce post harvest losses. To reduce post harvest losses. A very popular example is flower sour tomato. This tomato has an increased shelf life because of inactivation of the gene producing an enzyme called as polygalacturonase called as polygalacturonase. This polygalacturonase digests pectin in cell wall. So this was responsible for tomatoes going bad very fast. So what we did was we inactivated this gene, and this enzyme is no longer produced, and the shelf life of this tomato is increased. The third use is making crop plants more tolerant to abiotic stress such as cold, drought, salinity, acidity, etc. So these genetically modified plants, they can thrive over various environmental conditions such as cold, drought, and acidity, salinity, etc. The next use is to increase the efficiency of mineral usage by plants. To increase the efficiency of mineral usage by plants. This prevents early exhaustion of soil nutrients. So we can plant more crops on such soil which has not been deprived of nutrients. Next is to produce biopharmaceutical products. To produce biopharmaceutical products. A very good example is production of hirudin by Brassica napus which is the mustard plant. This hirudin, it acts as an anticoagulant or a blood thinner. Hirudin is naturally produced by hirudinaria called as leech. So what we did was we took the gene for hirudin production and put it in Brassica napus, and now this Brassica napus produces hirudin. It produces hirudin. This hirudin gets accumulated in seeds. From the seeds, it is extracted and it is purified, and then it is used in clinics or for research purposes. Next use is to produce herbicide or weedicide resistant plants. So no matter how strong of a herbicide or weedicide you spray in your field, these plants will not be affected. Example, glyphosate resistant tobacco. This tobacco was the first transgenic plant. So the first transgenic plant, or the plant whose genes have been manipulated, which was ever produced, was a tobacco plant. Next is pest resistant crops. Now the details of pest resistant crops we will discuss in the next video. With this, we finish off with this discussion on use of genetically modified organisms. See you in next video.